Botulism and the Beached Whale, a Clostridium botulinum story, written by Joy Shaw. Good morning, Toklo. What are you doing today? Oh, look what you do, Anna. After school, my friends and I are going to look at that beached whale down by the shore. You mean the beached whale that's been sitting on the beach for weeks? Yeah, my friend Philip said we should harvest it so it doesn't go to waste. Toklo, I don't think that is such a good idea. What? Why not? Aren't you always telling me not to waste food? Yes, of course, and we rely on whales and seals for survival, but I think that whale has been beached for far too long. Aren't you worried about botulism? Botulism? What's that? Oh, Toklo, you need to know about this. Come sit with me, and I'll tell you. There are bacteria in our environment, and even some within us, from a genus called Clostridium. There are some species, like C. difficile, that normally live in our gut and don't cause problems under normal circumstances. But there are some species that can become incredibly dangerous, like C. perfringens and C. tetani, the causative agent of tetanus. And there is one species that is especially dangerous for us. It is called Clostridium botulinum. It is a gram-positive, rod-shaped, motile, catalase-negative bacteria, and it's strictly anaerobic, which means that they like low-oxygen environments because oxygen will kill it. You're kind of losing me here. What are we talking about again? Okay, well, here's the part where you should pay attention. C. botulinum can grow fast, because they reproduce with binary fission. However, the main reason C. botulinum is so dangerous is because it produces something called endospores. When the bacteria is under a lot of stress, they hibernate by forming spores as a way to protect their DNA. And when the spores have the right conditions to grow again, like once they're in your gut, in a wound, or in food, they produce a life-threatening neurotoxin called botulinum toxin, which causes an infection called botulism. Botulinum toxin is one of the most lethal toxins on Earth. Even exposure to microscopic amounts can be deadly. The toxin inhibits the release of acetylcholine at the neuromuscular junction, so it can stop muscles from contracting. In only 12 to 36 hours after exposure, symptoms can start like nausea, vomiting, weakness, abdominal pain, diarrhea, constipation, and vertigo. And then comes the neurological symptoms like blurred and double vision, difficulty speaking and swallowing, fatigue, loss of muscle coordination, difficulty breathing, and even flaccid paralysis and death. Okay, that sounds super scary, alright, but what does that have to do with the beached whale? I'm getting there, Toklo. I promise. Okay, where was I? Oh yes, so there are 4 types of botulism: foodborne, wound, infant, and inhalation. And did you know that Alaska has the highest rate of foodborne botulism in the country? Wow, really? Yes, and most of it has traced back to the traditional foods that are staples in our diet as Native Alaskans. Seal flipper, whale muktuk, aged fish, beaver tail, and seal oil are frequent carriers of the botulinum toxin, especially when using methods like home canning or fermentation. There are many types of the botulinum neurotoxin, which range from type A through G, but only types A, B, E, and F cause illness in humans. However, the one that concerns us the most here in Alaska is type E. This is because type E toxin is exclusively found in animal foods of marine origin. Oh, I think I see where you're going with this now. Exactly. That whale, who has probably interacted with the C. botulinum spores that like to hang out in the soil and marine sediments, and has also been dead for far too long, is probably carrying the botulinum spores? Is definitely carrying the botulinum spores. C. botulinum is a saprophytic bacterium. This means it lives and breaks down decaying organic matter just like that beached whale. Ah, that's a shame. Well, I guess I'll just have to cook it first. No. Don't you remember what I said about endospores? They are formed to protect DNA. That means that regular cooking and processing methods don't kill spores because they are so tough. Oh my goodness, next you're gonna tell me I can't freeze it either? Correct. Freezing can only slow them down, not destroy them. Botulinum spores start producing toxins at only 39.2 degrees Fahrenheit. That's why scientists are worried about the potential for deadly outbreaks because of global warming in the Arctic. So, is there really nothing I can do with that whale? What a huge waste. Well, I personally wouldn't risk it, but if you do end up eating it, despite my warnings, I'll let you know how to prevent botulism. What? You know how to prevent botulism, but you still made me sit here and listen to your whole story? Yes. But that's because you needed to know how dangerous it is, and so that you can warn your friends and family. Okay, so here are some things you should remember. Since C. botulinum is an anaerobe, it thrives in low-oxygen places like canned goods. but it also can't grow in acidic foods under 4.6 pH, so high acid canned foods are generally safe to eat. If you're making and eating low-acid, home-processed foods like meats and vegetables, you must use the pressure canning method to reach 240 to 250 degrees Fahrenheit to kill the spores, and boil the cans in water for 10 minutes prior to eating to kill the toxin. Never eat from any can, home-processed or store-bought, that has been damaged, leaking, bulging, or that looks abnormal in any way. Never use cans that spurt liquid or foam when opened. Discard any questionable canned foods by double bagging and throwing it away outside of your home, away from people and animals. Some animals can get botulism too. Refrigerate all leftovers and cooked foods within 2 hours after cooking. And additives like sodium nitrates can help inhibit spore growth in cured meats. And if you ever suspect you or someone you know may be infected with botulism, get medical help immediately. Antibiotics can work to combat an overgrowth of C. botulinum vegetative cells, but only an antitoxin can work to neutralize toxin not yet bound to nerve endings. Alaska's Department of Health and Social Services keeps a stockpile of antitoxin kits in Anchorage, which are distributed to local health centers throughout the Alaskan bush. Okay, thanks, Anna. I'll tell Philip that our whale harvest is back on. No. I still wouldn't trust that particular whale.
This is my favorite microbe, Escherichia coli. E. coli lives in the gut of mammals, and most strains are beneficial, but there are some that are pathogenic. E. coli was originally discovered by Theodore Escherich in 1884. At that time, Escherich was working towards finding the cause for traveler's diarrhea and urinary tract infections. And E. coli is a gram-negative bacteria. The shape and anatomy of E. coli: it is a bacillus rod-shaped organism in a single-cell arrangement, and it reproduces using binary fission. And there can be virulence factors such as fimbriate and adhesion factors. The phylogeny is E. coli is a member of the Gamma Proteobacteria phylum, which is mainly gram-negative bacteria, part of the Enterobacteriaceae family as well. This family includes Klebsiella, Serratia, and Enterobacter. The nutrition, growth, and metabolism of E. coli. E. coli is a facultative anaerobe, which means it can grow with or without oxygen. It catalyzes carbohydrates via glycolysis and the pentose phosphate pathway, and it typically thrives in the human gut, aiding with digestion, breaking down complex carbohydrates. It also produces vitamin B12 and K for us. We have vitamin K and then vitamin B12. Interactions with other microorganisms. So E. coli works synergistically with other beneficial bacteria in the gut, such as Lactobacillus and Bifidobacterium. E. coli also produces short-chain fatty acids like acetate and propionate, which support gut health. It participates in cross-feeding with other microbes, exchanging nutrients like sugars and amino acids. And then it also forms biofilms in the gut, which helps enhance the stability of and protection of the gut. It helps the E. coli kind of take root in our gut so that the more opportunistic negative bacteria does not take hold. E. coli in the environment. It does thrive in the human gut, but it also, because it's found in the human gut, it is found in soil, because soil can start out as organic matter that passes through other larger organisms. It is also found decaying vegetation anywhere on the ground, and you can also find it in water. Uh, as it being a coliform, so it is found in the gut of mammals, it being present in water sources can indicate that it has been contaminated, potentially with poor sewage treatment or just any form of fecal contamination. The pathogenicity is more towards specific strains. One of those strains is going to be O157H7, which can cause serious illnesses, including bloody diarrhea and kidney failure. Most E. coli, or all E. coli, are going to be named for what they cause. So enterohemorrhagic E. coli causes bloody diarrhea and hemolytic uremic syndrome. Uropathogenic E. coli leads to urinary tract infections, and enteroinvasive E. coli causes severe diarrhea and high fever. So that was one of the common ones, but other common ones also include O111 and O26. And transmission of the O157H7 typically is going to happen from consuming undercooked ground beef, unpasteurized milk, or contaminated water. And then pathogenicity continued. Typically, the most common disease that you're going to find with E. coli is gastroenteritis, which is caused by enterotoxins that bind to the intestinal cells that are lining your intestines, which leads to diarrhea, cramps, vomiting, and then urinary tract infections are caused by E. coli being transferred from a fecal area to the urethra, and it is typically seen more in humans who have a vagina because the urethra is closer to the anus. It is also seen for neonatal meningitis to be caused by E. coli, as well as sepsis if E. coli makes its way into your blood. E. coli is not all bad. It does also have several medical and biotechnical advancements. We recently saw one with the gene of bioluminescence being added to the E. coli. There are other useful methods for this, some of these being for insulin being produced. This provides a safer and more efficient way to produce the insulin and harvest it. Other compounds that are being produced from using DNA recombinant technology with E. coli would be for interleukins, which are meant to enhance immunity, tumor necrosis factors, these are for helping cancer therapy, interferons for multiple diseases. There's a very large list of those, which include multiple sclerosis, and then macrophage colony stimulating factor. This will help stimulate immune cell production. This is good for patients who are going through heavy chemotherapy, which is suppressing their immune system. And it also helps produce vaccines and other biopharmaceuticals. And that was E. coli.
Hello everyone. I hope you guys had as much of a fun time working on these term projects as I did. Today I'm going to talk about my microorganism of choice, Rhizobium. This is a rod-shaped genus of bacteria that is primarily responsible for nitrogen fixation in plants. First things first, I'd like to start with some background information. We'll start with the phylogeny of Rhizobium. It comes from the family Rhizobiaceae, the order Rhizobialis, belongs to the class Alpha-Proteobacteria, and the phylum Pseudomonodonta, also known as Proteobacteria. Now, Rhizobium is essential for providing nitrogen fixation to a variety of flowering plants and species of legumes. Legumes include peas, beans, clovers, and a couple of other things. Next, I wanted to evaluate the importance of nitrogen fixation by Rhizobium. Nitrogen is essential for many things within our own body, including DNA and RNA synthesis. It's also important for the creation of amino acids, and therefore it's important for building proteins. Now, you might think, well, nitrogen's such a big part of our atmosphere. Well, atmospheric nitrogen in the form of nitrogen gas, N2, which makes up over 70% of our atmosphere, is actually unable to be used by plants and animals due to its strong polar covalent bonds, so it must be reduced to a usable form of nitrogen through nitrogen fixing or nitrifying bacteria. Now here in this slide, we'll go over the actual process of nitrogen fixation performed by the Rhizobium genus of bacteria. The process of nitrogen fixation takes a total of 16 adenosine triphosphate, or ATP molecules, in order to cleave the triple covalent bond of nitrogen gas. That's an extremely strong covalent bond, and that's why we need this process. These ATP that we use come from metabolizing carbohydrates that the plant, where the Rhizobium bacteria reside, produce during photosynthesis. Passes through root nodules, which we'll go over in the next slide, in exchange for the end product of nitrogen fixation, ammonia. So bacteria receive nutrients, plant receives ammonia, a usable form of nitrogen. Rhizobium receives most of its essential nutrients for metabolism and cellular function through this symbiotic exchange. I also have here the full chemical equation for nitrogen fixation. Here on this slide, we have root nodules and the rhizosphere. A root nodule is a secondary structure produced by certain plants, mostly the legumes and the flowering plants that host Rhizobium, the genus that we're discussing. They host these bacteria, these nitrogen-fixing bacteria, in order to better form their symbiotic relationship and exchange nutrients. Now, a lot of plants don't have root nodules, so they have what's called a rhizosphere, which is the area of soil around a root that the plant directly can influence the chemistry of, leading to high concentrations of nitrogen-fixing Rhizobium, as well as other nitrogen-fixing bacteria or nitrifying bacteria that become concentrated in this close area around a host plant. While that's much of the importance of Rhizobium as a genus nitrogen fixation, I also wanted to bring up something pretty interesting that's been happening in the scientific world. Here we have some of the first pictures of what's called a nitroplast, and I'll read through this for you guys. A team at UC Santa Cruz has finally isolated the first ever nitroplast organelle. Normally, nitrogen fixation, as we talked about, is only able to be performed in the method described. However, the development of a nitrogen-fixing organelle within a eukaryotic cell, it was an algae cell that adopted a cyanobacteria, was discovered in an event of endosymbiosis. A eukaryotic cell was able to engulf a symbiotic prokaryotic cell and replicate it into consistent organelles throughout generations. Now, the last couple times this happened, we got mitochondria and we got chloroplasts. These were world-changing organelles, so it's interesting to see something new developing over the past couple million years that we finally discovered real cases of. Thank you for listening. I hope you guys enjoyed and hopefully learned something. Thank you so much.
The French, Polish, Spanish, Neapolitan, and Christian diseases. Treponema pallidum, i.e. syphilis. Syphilis was first documented in 15th century Naples during the first of the Italian Wars. Where it came from, who's responsible, and what it even was up to debate until Fritz Schnauden and Eric Hoffman in 1905 discovered the culprit. Treponema pallidum. Syphilis plagued the world uninterrupted for centuries, seeing major booms in times of conflict. Regions and countries all had their own unique names for the infection. The Italians called it the French disease, the Turks called it the Christian disease, and the Dutch called it the Spanish disease. A classical case of propaganda used to other, scapegoat, or ostracize a group of people or other nations. Though arguments persisted on who to blame, one thing was clear. Syphilis, most commonly, was transmitted sexually. This caused some problems for people in society who were supposedly celibate. Most notably, Pope Julius II contracted the disease. Let's get on with it. What is T. pallidum? T. pallidum is a gram-negative, spirochete in shape bacterium that causes infection primarily through sexual transmission. T. pallidum has limited metabolic capabilities with an unusually small genome and lacks many genes that code for metabolic functions and virulence factors. It cannot survive outside of the human host. Current circulation of global syphilis is comprised of two branching lineages, being SS14 in the Nichols-like group. There are 4 stages of syphilis, each with their own unique characteristics. During the primary stage, T. pallidum penetrates intact mucous membranes or dermal abrasions. A single sore, known as a chancre, will appear at the site of infection. Areas can include the penis, vagina, anus, rectum, and lips or mouth. Typically, the sores are painless, firm and round, appearing red in color. At this stage, contagion risk is high, and because the chancre is usually painless, it commonly goes unnoticed. Without treatment, it will go away within a few weeks to months, but this does not mean it's gone. Syphilis will then move on to the second stage. Hey, here's Pope Julius II again. He's represented here as secondary syphilis, a rash that can cover the entire body, including the bottom of the feet or palms. It may flare up or subside for years without treatment. Transmission during this period can again be through vaginal, anal, oral, or direct contact with an open sore. Other symptoms may include swollen lymph nodes, fever, and fatigue. In the latent stage of syphilis, there are minimal outward signs of the ongoing infection. This phase can last up to 20 years with flare-ups from time to time. Transmission is rare. About 30% of people progress into the final stage, also known as the tertiary stage, which can cause a range of complications, including damage to the brain, eyes, nerves, heart, liver, blood vessels, and bones. Another pathway of syphilis infection is congenital, originating from the bloodstream of an infected mother to the developing fetus. Without antibiotic treatment within the first 2 trimesters, this can result in fetal damage, stillbirth, premature delivery, or fetal death. Severity of effects are suspected to depend on fetal immune response. Now let's wrap this up with a fun fact and suggestions on further readings. The use of condoms exploded in the 1500s in thanks to the response to the rapid outbreaks of syphilis. I also highly suggest reading about the Tuskegee experiments. In an attempt to study the impacts of syphilis throughout a human's lifespan, black men were unwilling participants. Between 1932 and 1947, about 600 men were lied to about their syphilis status and not given treatment. At least 128 of these men died, many after penicillin became the primary treatment. This dark moment in medical history is in part why we have informed consent laws today. Thanks for listening or reading.
Good afternoon, everyone. Today we are diving into the world of a rather sneaky bacterium, Actinomyces israelii. This microorganism quietly resides in our mouths, but can cause significant oral infections when given the opportunity. As future dental professionals, understanding this hidden threat is crucial for effectively managing oral health. We will explore its biology, clinical significance, and how it impacts our daily practice. Actinomyces israelii was first identified by James Israel, a German surgeon, in the 19th century. It was initially mistaken for a fungus because of its branching, filamentous structure, which gives it a fungal-like appearance. Later, scientists realized it was actually a bacterium, leading to its classification under the genus Actinomyces. In the oral cavity, it usually resides as a harmless commensal organism. However, it can become an opportunistic pathogen when mucosal barriers are compromised, such as during dental procedures or trauma. This dual nature makes it both fascinating and clinically significant for dental practitioners. Anatomically, A. israeli is quite unique. It is a gram-positive filamentous bacterium with a branching morphology, often mistaken for fungi. It exhibits pleomorphism, appearing as filaments, rods, or coccoid forms based on environmental conditions. Its thick peptidoglycan cell wall provides structural integrity, while lipoteichoic acids enhance adhesion, promoting biofilm formation. A. israeli thrives in anaerobic environments, like between the periodontal ligaments, as shown on the right on the clay tooth sculpture, dental plaque, and tonsillar crypts. Normally harmless, it becomes pathogenic when mucosal barriers are compromised, leading to chronic infections and abscess formation. A. israelii belongs to the phylum Actinobacteria, known for its high G plus cDNA content, which enhances its environmental resilience. It is closely related to other Actinomyces species like A. neslundi, A. viscosus, and A. odontolyticus, all of which are involved in oral health and disease. It also shares ancestry with Nocardia and Propionibacterium, highlighting its evolutionary adaptability. Interestingly, it is genetically linked to Streptomyces, known for producing antibiotics. This evolutionary background underscores its genetic versatility, making it a significant player in both microbial stability and oral infections. Actinomyces israelii is a chemoorgan autotroph, fermenting glucose, fructose, and lactose to produce lactic and acetic acids, creating acidic dental plaque that inhibits less acid-tolerant bacteria. The diagram shows its metabolic flexibility. Following the Embden-Meyerhoff-Parnas pathway, it undergoes anaerobic fermentation, producing acids, or aerobic respiration via the pentose phosphate pathway when oxygen is available. This allows it to thrive in both oxygen-poor gum pockets and oxygen-rich enamel surfaces. The pH scale highlights how fermentation lowers pH, enhancing microbial growth and biofilm formation in the oral cavity. Actinomyces israelii is a commensal bacterium in the oral cavity, but can become opportunistic when mucosal barriers are compromised. It causes cervicofacial actinomycosis, leading to chronic abscesses and sulfur granules that shield it from immune defenses. It is frequently associated with dental procedures like root canals and extractions, resulting in persistent infections. Beyond the oral cavity, A. israelii can spread systemically, affecting the lungs, chronic cough, chest pain, gastrointestinal tract, abdominal abscesses, fever, and central nervous system, brain abscesses, neurological issues. Emerging evidence also links it to cardiovascular disease via chronic inflammation. Recognizing its pathogenic potential is crucial for early diagnosis, treatment, and prevention in dental practice. Actinomyces israelii infections are often misdiagnosed because of their slow progression and resistance to conventional treatments. This case highlights how persistent periapical infections may signal something more serious: apical actinomycosis. A 24-year-old female presented with persistent swelling and dull pain in the mandibular region, despite having undergone root canal treatment on teeth 30 and 31. Radiographs revealed a large periapical lesion, suggesting a chronic infection. Since typical endodontic treatments had failed, an apicoectomy with curettage was performed. Intraoperatively, yellow sulfur granules were observed, a hallmark of A. israelii. A biopsy confirmed actinomycosis, requiring a 7-day course of high-dose amoxicillin, 500 milligrams every 8 hours, to eradicate the infection. At the 2-year follow-up, the patient remained asymptomatic, and radiographs showed partial healing. This case reinforces the importance of recognizing actinomycosis as a cause of persistent periapical lesions, particularly in infections that do not respond to standard endodontic therapy. As future dentists, you play a key role in preventing A. israelii infections. It thrives in poor hygiene, deep biofilms, and untreated infections. Prevention requires early detection, plaque removal, and proper management of trauma, root canals, and extractions. Recognizing persistent infections early ensures timely debridement and targeted antibiotics. By prioritizing preventive care, patient education, and microbial balance, you can stop this pathogen before it strikes. Your expertise will keep your future patients safe from oral infections. Thank you.